Hi there, welcome to day 3 of the Neuro AI course. I'm Heiko Schütt and I'll guide you through this day together with Nico Kriegskorte, who you'll see later in this video. So today's talk is about the big question, what does it mean to say that one neural network is like another one? And here neural network could be a biological one, i.e. a brain, or a deep neural network. And it should be immediately clear that this is an interesting question. For neuroscience, this is how you test whether your model is any good, right? You want to compare it to the brain and want to say whether it is like the brain or not. And in machine learning, it is a great question to ask yourself for understanding deep neural networks to first understand which of those are actually similar to each other and which ones are different. So we are spending a whole day on this question because it is actually a hard one. And it is a hard one because deep neural networks are very different from each other. This is illustrated here on the slide for this very simple 3-layer recurrent neural network on top and a mixer architecture of which you see only a single layer here. And as you might notice, despite those networks both are processing images and perhaps doing a classification task on them or something like that, there's really nothing in those networks that maps from one to the other, right? So we have no idea whether any of those channels here or patches or MLPs or something in any way match to anything to the network on top. And this makes comparisons hard, right? So making any judgments about whether those two networks are similar or not are really hard. Nonetheless, one thing we can do for any deep neural network is to convert it to its computational graph, right? So that means that we are splitting things into representations. In the deep neural network packages, those are called tensors, right? And then there are functions or transformations that compute the later representations from the previous ones. And this we can do for any deep neural network. And for biological networks, we can think of it in similar ways, except that we usually want to include time, so that the same brain area could have different representations at different time points, and later time points can be computed based on any earlier time points. So before I explain to you how we actually compare those things, let's talk a little bit about other ways one could think of that are rarely used. First, you can think about, well, we can just tell them what kind of architecture it is, and that should be really informative. And unfortunately, that's not true, right? Different neural networks with very different architectures can basically implement the same functions, such that they should be considered exactly equivalent, but they have completely different architectures. And additionally, it is by all standards a very coarse distinction, right? If you just know it is a transformer or it is a convolutional neural network, that tells you very little about what the neural network is actually doing. And if you train on different tasks or different outputs, you might also, for the same architecture, get very different representations and very different behavior of the neural network. So that doesn't work too well. Then we can talk about connectivity. So that's an idea that immediately comes up because what we just saw definitely is in the math sense a graph, right? Nodes connected by connections. But to make this meaningful, you really have to think that the different arrows are similar in meaning. And this is not even true within one network in most cases, and in definitely not across networks. So different networks will use completely different computations, and then two nodes being connected means entirely different things. And that breaks basically all the assumptions of graph theory. And that's why this is only applied if you are talking about big bunches of neurons that are all the same and computed in the same way. Then this can be sensible, but it is a very restricted set of comparisons you can make. And in some ways, weights is even worse. So if you want to compare the weights from one network to another, you not only need it to be the same kind of function so that the weights have any like meaningful way of being compared, but also you want that the representations you come from and the representations you go to already are similar in meaning. And this restricts you to like essentially identical networks in terms of architecture for your comparisons. So what can you do if you want to compare networks in general? Well, one thing you can do is to compare outputs. So the convenient thing is if you have two networks and they are trained for the same task, they will produce output in the same format. So you can compare them, right? And the main thing I want to say about this approach here is that you can do more than just comparing how well they do on the task, right? You can also analyze on what images do they fail and fail in what ways. So if you're talking about classification, for example, what wrong class do they put the stimuli in and things like this. And this has been done very successfully. And the other part is that you don't need to restrict yourself to natural, normal images. You can also create other images for testing. And this again has been done very successfully. Think of adversarial examples here, or in the Golan papers I'm citing here, it's about controversial stimuli. So stimuli that are explicitly created to make the networks do different things. And so you can do this, and this is done a lot. But of course, when we are talking only about outputs, we are not using any of the information about the transformation we do beforehand, right? And in neuroscience, we kind of hope that measuring the brain helps us understand it. And in similar ways, in machine learning, having access to all the intermediate representations should somehow help us understand the deep neural networks better, right? So how do we do that? And in most cases, as we've seen, the functions are really hard to compare. So what we usually do is just compare representations. Yeah. This is what we'll do to understand the internal processing of deep neural networks. And here, the aim is essentially to find kind of metric space of representations, right? So if we have two networks with multiple intermediate representations, we want to find a way to measure the similarity of each pair of representations such that we can embed them into a space where, how far they are apart corresponds to how similar they are, right? And then because we know which of those points corresponds to which representation, we can talk about things like how much does each of the computations transform the representation, which of the representations are close to each other, i.e. somewhat corresponding, and then also about whole paths here, which then correspond to the full networks, saying something like, these two networks appear to be doing something very similar. And again, for that, we need now comparability of any of the arrows. We just need a comparison between those representations. And with that, we have now reduced the problem to comparing representations. So what is a representation concretely here is we have a bunch of test stimuli. We're talking here about images. It would work similarly if those were text snippets or similar things. And then we have a representation, so just the activations for all the neurons in one of those representations, one of the layers. And we have this for a second representation. They don't need to have equally many neurons, right? They might have different amounts of them. And now we need to find some way of comparing them to each other. And now I hand it off to Nico, who will tell you how to do that.
All right, let's talk about comparing representations in terms of features or representational geometry. So we're going to talk about two approaches to comparing representations. Both of these are very important in computational neuroscience. The first one is linear encoding models, where we fit linear mappings between our model, like a neural network model, and the recorded responses of neurons. And the second one is representational similarity analysis, where we first compute representational distance matrices for both the model and a brain representation, and then compare these representational distance matrices. So let's look at this in a little more detail. So as Heiko has shown you, we might have a number of visual stimuli or other inputs to the systems that we're looking to compare in terms of their internal representations. And these inputs are presented to each of our systems, for example, to brains of subjects in an experiment. And for each of them, we get an activity pattern, for example, in a cortical area. So this gives us a matrix indexed vertically by the stimuli and horizontally by the neurons, where each row is an activity pattern and each column is what we call an activity profile. So this is a pattern for one neuron of the response of that one neuron across all the different stimuli. Sometimes these are also called tuning curves of neurons. So these characterize the response cell activities of particular neurons. We present the same stimuli to the other system. So in this case, this could be a layer in a deep neural network model. And again, we get a pattern for each of the particular stimuli. And again, we get this kind of response matrix, stimuli by neurons. So one thing that we note immediately about these two matrices is that they are for the same stimuli, so they have the same height, but they have different widths in general. And that's because our two systems might not have the same number of neurons in the representations that we're looking to compare. So this makes it difficult to pursue a kind of simple comparison of one activity profile to another activity profile. We will not have a one-to-one correspondency mapping here. We don't even have the same number of neurons. So one approach is to fit linear encoding models, where we fit a linear mapping from each of the neurons of our model layer to each of our measured neurons in the brain region that we're looking at. And this is a directed model. So the goal is to predict the brain responses from the neural network responses. And that's natural in this kind of comparison, where we have measured data on one side from the brain, and we have a noise-free model on the other side that we wish to evaluate. However, if we had a situation where there's more of a symmetry, where both of the systems we're comparing are neural networks and there is no notion of noise, then we'd have to make a decision here in which direction we fit the linear encoding model. And since this comparison is fundamentally asymmetric, the results might not be identical. And it might be interesting for you to think about in what kinds of scenarios the results of fitting the model in one direction or the other direction might differ. So some important facts about this approach to comparing representations is that the comparison is fundamentally directed, and that we need to fit these n1 by n2 parameters, where n1 is the number of neurons in the model layer and n2 is the number of neurons in the brain region. So we need a separate training set with different stimuli in order to fit those parameters, and then we can evaluate on a different set of stimuli for which we've also measured responses in both systems, how well this mapping performs. Another approach to this problem is to first compute the distances for each pair of response patterns. So we take, for example, the first two response patterns here. We compute the distance between them using either just a distance function or some estimator of the distance that we're going to talk about more later. And this gives us a matrix, a square matrix, indexed horizontally and vertically by the stimuli, which gives us the distance for each pair of stimuli between the response patterns in the multivariate neural response space. We can do the same thing for the other representation, and we get another square matrix indexed vertically and horizontally by the stimuli. And now the colors here encode no longer the activity, but the dissimilarity or the distance. We use the term dissimilarity as a more general concept that includes distance estimators that are not themselves distances. So on this basis, we can directly compare the representations because they're now square matrices with the same dimensions, and they're indexed by the stimuli, so they have matching rows and columns. And this gives us a way to symmetrically compare the two representations. For example, we could choose some symmetric comparator, like a correlation coefficient, or we could compute a simple distance like the Euclidean distance or the angle between the two representational dissimilarity matrices as vectors in a space of RDNs. So this is a zero parameter comparison that's possible here, and any fitting of model parameters is optional. So we don't have to fit parameters just to compare two representations. Although, of course, we could have parameters in the model that we adjust so as to make the model have a representation that's as similar as possible to the brain representation. So in tutorial 2, you'll think more about computation as the transformation of representational geometries and about this concept of a metric space of representations that Heiko has already introduced you to. And there would be critical to have these symmetric measures that enable us to compare different representations, even when they have different numbers of neurons. So now that we have kind of a sense of the lay of the land and how these two approaches differ, let's dig a little bit deeper and let's look at this response matrix that we call R in a bit more detail. So I've told you that we can think of this in terms of its rows, the activity patterns, or in terms of its columns, the activity profiles. Let's consider the columns first. Each of the columns is a point in a high-dimensional space where each dimension corresponds to a stimulus. So we have the first neuron's activity profile or tuning curve, and the tuning curve, the values of the tuning curve are the coordinates of this point in the space spanned by the stimuli. And for each of the neurons, we have a point here corresponding to one of the columns of the matrix R, the response matrix. And when we model the representation, we have a basis set of activity profiles. And our goal is to find a set of weights W such that if we combine the model's activity profiles with the weights W, we recover the brain response matrix as well as possible. And usually we have more predictor variables, more activity profiles. So this is an ill-posed problem, and we need some kind of prior on the weights for this linear mapping problem. And the most popular choice there is to put an L2 penalty on the weights, equivalent to a Gaussian isotropic prior on the weights. And that's called ridge regression. So this is the approach of linear encoding models in a little more detail. Alternatively, we can think of the stimulus response matrix in terms of its rows, the activity patterns. Each of the activity patterns lives in a multivariate space again, and that's the neural multivariate response space. So here, each dimension is a neuron, and a point here corresponds to an activity pattern elicited by one particular stimulus. So we can look at where these patterns of response elicited by the different stimuli fall, and each of them corresponds to one of the rows. And once we have the points in the space, we can look at their pairwise distances to characterize the geometry of these response patterns in the multivariate response space. And we can characterize this geometry by the distance matrix D, and that's the approach we take in representational similarity analysis. There's also a third approach which links these two approaches in a very interesting way and is therefore important to consider to understand the relationship between linear encoding models and representational similarity analysis better. In this approach, we consider the space spanned by the stimuli. So we're looking at the distribution of points here, where each point is an activity profile. And our goal is to model this distribution of activity profiles. And what we do is we just model this as a multi-normal distribution in the space spanned by the stimuli. So the multinormal distribution, a Gaussian distribution, is characterized by its sufficient statistic, the second moment matrix G of the activity profiles. And it turns out that G and D, the distance matrix, contain the same information. So we can convert one into the other and go back and forth. The only thing we need for that is to include the baseline pattern, the origin, as another pattern in the distance matrix. The second moment matrix is a similarity matrix, and the distance matrix is a dissimilarity matrix. So it's perhaps intuitive at a very high level that if you know the similarities, you can infer the dissimilarities and vice versa. So the second moment is closely related to the Gram matrix, the inner products of the activity patterns, so how similar they are, only it's normalized. So we can think of it as a covariance matrix that defines this multinormal distribution. Using this for inference on models is called pattern component modeling, and it's a very powerful approach that's been introduced by W1D3. So these two on the left here have an important commonality. They model the activity profiles distribution, and they consider the space spanned by the stimuli. By contrast, representational similarity analysis models the representational distances. So models predict distance matrices. And these two on the right here have an important commonality as well, which is that they use a stimulus by stimulus square matrix of summary statistics to characterize the representation. Only in the case of the pattern component model, it's the second moment matrix or a similarity matrix. And in the case of representational similarity analysis, it's the distance matrix. But we've seen that these contain the same information. By contrast, in linear encoding models, we model each response separately. So it's a mass univariate approach where we go through all the neurons measured in the brain and model each as a linear combination of the model neurons.
Okay, hi. So Nico just told you that most of the methods we use for comparing representations are based on the similarity structure of the stimuli with each other. So which stimuli have similar representations to which others? And I will now spend the rest of my time I have with you trying to convince you that this is indeed sensible, so that it is a good idea to compare representations based on this similarity structure. The first step towards doing this is showing you that it doesn't really matter whether we're talking about the similarity matrix, which is also known as the second moment matrix, Gram matrix, or linear kernel matrix, or about the dissimilarity matrix or distance matrix. And first step for doing this is a pair for the same data, similarity matrix and dissimilarity matrix, and you should immediately see that they are closely related to each other. To make this mathematically more rigorous, we can write down the formulas for each of those matrices. For the left one, it is just the inner product, so Ri times Rj. For the distances, it's the difference between those representations multiplied with itself. And now we can immediately just multiply out this formula for the distances and get that the squared distances is a simple linear sum of the entries from the kernel matrix. So there is a linear map from the kernel matrix to the distance matrix. Now, very attentive listeners might notice that there is diagonal of zeros in the distance matrix. So the distance matrix has a few dimensions fewer than the kernel matrix. We can fix that, however, by adding one more row and one more column to the dissimilarity matrix, which is the distance to zero. And this distance to zero is immediately the diagonal of the kernel matrix, which you can see by just plugging in 0 here for Rj, and then see that this is then the inner product of Ri with itself. So the distance to 0 is this diagonal. And once you do that, the distance matrix is completely equivalent to the similarity matrix as it is related to it by an invertible linear transform, making them about as similar as they could ever be. So now that I've shown you that they are kind of the same thing, why should you use either of them? To make this argument, I'll start by showing you the relationship between similarity and generalization of readouts from the representation. This also nicely ties our day to the other days of the Neuro AI course, where you heard a lot about generalization. So let's have a look at how that looks like. So what is the generalization I'm talking about here? We have some input images here, which we first project into the same representation, and then we have a linear readout to some kind of task that produces those numbers. And now we want to understand how the setting of training data we have will lead to predictions of the test data, right? And the different types of generalization you would have heard about so far would correspond to what kind of images do you actually use here, right? So if those come from a different distribution than the test data, then this would be out of distribution generalization, for example. So to understand this more, let's have a look at this linear regression step. Again, we're talking about linear regression from our representation X, which is stimulus where wise each a vector of features. And we will make a linear model to our training labels or outputs by adding a vector of weights B, one for each feature. And this will give us our map to go make our predictions. We can fit this by just ordinary least squares. So we just minimize the square difference between the actual data and our prediction. And this will give us a vector of weights here. And then we can take our weight vector, multiply it with the features for our test stimuli, and make predictions, right? So this is how we would train a linear readout from our model. To understand how this linear readout generalizes, we need to put those two things together, which gives us this formula, and then look at how we get actually from the representations of our training and test stimuli and our training data to some prediction. Usually we would think of this as a map from our representations of the test stimuli to an output, and those are the weights we get. For generalization, a different linear map is much more interesting, which we get by just moving those brackets one to the left, and then noticing that with this, we now have a linear map from our training data to our test data. So there is a set of weights determined by the representation, how to combine our training data to get the test data. And now, to understand how the generalization actually happens, we should look at what those weights actually are. So let's do that. First of all, this is something which here on the left has the test stimuli and here on the right has the training stimuli. And there is a square matrix here in the middle. Thinking of this X transpose X as a kind of covariance formula, we can call this inverse here a precision matrix lambda, which simplifies the formula. And then we can see that this is indeed an inner product, right? So this is X transpose a positive definite matrix times X, which is a form called inner product. And that is a form of similarity. If this doesn't immediately make sense to you, perhaps this helps. You can decompose lambda into W transpose times W, and then you can see that W times all the x's projects your representation into a space where this formula here is the standard inner product. So again, highlighting this is a form of similarity, right? So we have a row vector of similarities of the test example to the training examples gives you the weights for making predictions. And now that we have this inner product, we can also make a matrix of all the inner products, which we then call the kernel matrix, right? So we just add this lambda to Ri transpose Rj, extending this slightly, and we also get a form of distance, which is again Ri minus Rj, and then applying the inner product of that difference with itself. And once we have this kind of lambda positive definite matrix here in there, we call that squared Mahalanobis distance. Yeah. So with this, we have now created similarity and dissimilarity matrices based on the linear readout, and they characterize the generalization. So how strongly a readout generalizes from one stimulus to another is a notion of similarity, making this a great basis for comparisons. And now I have three more reasons why similarities are a good thing for making comparisons between representations. First, they describe discriminability and decoding. So if we assume that our representations are not noise-free, but come with some error around them, then the distance will tell you how well those two can be discriminated from each other. The simplest case of that is if you look at isotropic Gaussian noise, then it is just a normal Euclidean distance between the two that determines how well you can discriminate those two patterns from each other. If you have correlated Gaussians, then you can use a Mahalanobis distance that we just got to know to make comparisons, and then you will see that, for example, the two ones here connected by the yellow ones would be further apart according to this Mahalanobis distance than the blue ones. And you'll see this in somewhat more detail in the tutorials. And then if there are other forms of noise that are not Gaussian, you'll not end up with Euclidean distances or those Mahalanobis distances, but more complicated ones. But there is always one, because if in doubt, you can always use a distance between those probability distributions as your distance measure. And then you would always keep discriminability. So how similar two stimuli are gives you how well you can discriminate them based on a readout. Now, the next point is that we know really well what the invariances are. So which representations end up being equal or equivalent according our distance measure. And concretely, those are if you're shifting everything in the representation, so adding something to this doesn't change any of the distances, and we are invariant to this. Rotations, so this is a special kind of redistributing the activity without changing the overall amount of activity. These measures are invariant to, and reflections too. So not only rotations, but also flips of the representation. And so those are the main three. And then we additionally ignore scaling usually. So if once we are actually computed the dissimilarity matrices, we use ways of comparing them that ignore how big the distances are overall, which makes us also invariant to just scaling everything up or everything down. And that's it. Yeah. Any other transformation to the representation will actually lead to different distances. So this is very nice because we really know which transformations we can apply without changing in representation space. And the third and last point I want to make is that distance are nicely preserved under random projections. This is known as the Johnson-Lindenstrauss lemma. So the idea is our neuronal representations live in a very, very high dimensional space, both for deep neural networks and in the brain. And we will often take a random projection down into much fewer measurements. For neuroscience, this is something like an idealized idea of what fMRI or EEG and so on might do. For deep neural networks, it's more of an idea we can effectively reduce the dimensionality of those representations substantially without changing the distances too much. So what does this lemma actually say? It says that if we have more than just a logarithmically growing bound on the number of points, then our random projections will yield only small distortions of the distances among points, independent of the representational dimensionality that we start with. Right? So this implies first that the expected distance after the projection is correct under some small, like mild conditions. The variance of the expected error is small, right? So we can actually use relatively few dimensions and still have a good estimate of this. And I want to note that this is the basis of a whole field in really big data science. So if you are talking about representations with really many dimensions, then it can be an interesting idea to apply these kind of random projections first and then compute only with data after this to bring everything into a manageable dimensionality. And for all of those the distance are well preserved. And of course, this also gives us hope for neuroscience that we might actually be able to measure those distances accurately enough to make sensible comparisons between the brain and neural networks. So with that, I hope I convinced you that similarities between stimuli are indeed a good basis for comparing representations. And we'll hand over to Nico again, who will tell you more details on which similarities should you actually be using, how do you deal with noise, etc. Have fun and see you at the tutorials.
Okay, thank you, Heiko. So in the last part of this lecture, we'll talk about the steps of representational similarity analysis one by one. The first step is to choose and use an RDM estimator to actually get an RDM, a representational dissimilarity matrix, from our data. The second step is to choose an RDM comparator in order to quantify the similarity between different representations. And the third step is model comparative inference, where we compare different models as models of a representation in a brain region so that we can learn something about the computational mechanisms underlying the representations in biological brain. So let's start with step one, estimating representational geometries by computing an RDM. We have to choose a distance estimator. So first, let's quickly recap what Heiko has already told you. We need to take correlated response pattern noise into account to make distances reflect the discriminability or the decoding accuracy for a pair of stimuli. If we use the squared Euclidean distance naively, we could have this case where we have a large Euclidean distance, but displacement between the two patterns is in a direction in which the noise is very large as well. So the stimuli are actually quite confusable. If you were given a response pattern from one of these two distributions and you were supposed to tell what the stimulus was, you'd make a lot of errors because these distributions are very overlapping. And even though this is the longest of the three Euclidean distances here, it's also the most confusable pair in this simple example. And as Heiko mentioned, a remedy for this is to whiten the space by applying a linear transform that renders these Gaussian distributions isotropic. And after doing this, we can use the Euclidean distance. And when we use the Euclidean distance after this whitening linear transform, that's actually equivalent to a different kind of distance, which is called the Mahalanobis distance, where you have the inverse of the error covariance here in between these two pattern differences, and that achieves this whitening transform. The second important consideration in estimating distances is that distance estimates from noisy data are positively biased. So one very quick way to understand this is to think of the case where two stimuli elicit exactly the same true activity pattern. So the true distance is 0, but now we get these noisy estimates of both patterns. And when we compute, for example, the Euclidean distance, of course, by definition, that's positive. So we're always going to overestimate this truly 0 distance. And that's this positive bias. And this positive bias affects distance estimates in general when we naively apply distances in order to estimate distances from noisy data. So let's think this through in a little more detail. We have two response patterns here, Ri and Rj, and these are the true noise-free response patterns. So we would like to estimate this true distance, the length of this gray line, Dij. However, we don't have access to these true response patterns. We just have measured brain activity data. So we have estimates that are affected by noise. And in a high-dimensional space, if the noise is isotropic, I'm assuming that here just to illustrate this, these error displacements that our pattern estimates are affected by will be approximately orthogonal to each other and also orthogonal to the connection line between the two true patterns. So in this scenario here, where we get a distance estimate from our pattern estimates that is longer than the true distance, and it's longer by a very predictable amount. So it's just since these elements are approximately orthogonal to each other, the norms of these errors squared and the true distance squared just add up to give us our squared Euclidean distance estimate. Here I've simulated this. I generated some random patterns of different true distances. Then I added noise, and I'm plotting the true distance, so the Euclidean distance without the noise versus the Euclidean distance with the noise. And unsurprisingly, we see this positive bias. So all of the points are above the identity line here. And it's particularly serious for the case when the true distance is very small. Note that here what I'm plotting is actually the distances, not the squared distances. If I plotted the squared distances, then you'd see just a shift and you wouldn't see this nonlinear effect. So here's another example where we have different true distances and in different shades of gray. And you can see that we only get the right estimate with a naive distance estimator, here the squared Euclidean distance, when the noise is 0. And as the noise goes up, we get a stronger and stronger inflation of the estimated distance. So what do we do? Well, we can remove the distance estimation bias. And how do we do that? We can use different data sets for these pattern differences before and after the inverse of the noise covariance. And to understand this a little bit better, we can remember that the first part of the definition of the Mahalanobis distance here is also the definition of the Fisher linear discriminant. So this is a vector W that we can project the data on in order to distinguish these two distributions of response patterns efficiently. And what we do in the so-called cross-Nobis distance estimator, which is short for cross-validated Mahalanobis distance estimator, is we take a separate data set and we have estimates for the two response patterns, Ri and Rj, from the separate test data set, and we project them onto the Fisher linear discriminant estimated with our first data set. And this is a way of removing the bias. When we plot the estimated distances as a function of the noise, you see for different true distances shown in different shades of gray here, you get on average the true distance. Even though, of course, as the noise goes up, the variability becomes larger and larger. There's nothing we can do about that. If there's more noise in the data, our estimates will be more variable, but on average, they'll be correct, and that's the definition of unbiasedness. So in tutorial 4, you'll play around with different ways of estimating distances. You'll learn about the Mahalanobis distance and how to whiten the space, and you'll also learn about removing the bias inherent to naive distance estimates. So let's move on to step 2 of RSA, which is choosing and applying an RDM comparator. So this is about quantifying the similarity of representational geometries by comparing different RDMs. The question is, what do you compute? How do you quantify how similar two RDMs are? An important issue in this context is that we need to account for dependency among dissimilarity estimates by whitening. Now, this might seem familiar. We just talked about whitening in the context of response pattern variability, noise in the space of the neural responses. Now we're talking about noise dependency in the space of RDM estimates. So we're talking about the space that the RDMs live in. So this space has one axis for each dissimilarity, so one axis per pair of stimuli. And a point in this space is an RDM. So here's an example where we have a data RDM D and two model RDMs M1 and M2. And if we naively selected the model that's closer to the data in terms of Euclidean distance, we would select model M2 here because it's closer in a Euclidean distance sense to the data RDM. However, when we consider the covariance of the RDM estimation error, we see that model 1 actually has the higher likelihood. So the probability of the data given model 1 is higher than the probability of the data given model 2 because the variability of the uncertainty about the actual RDM given a model is not isotropic. We can deal with this in the same way as at the level of the neural responses by whitening. So we can perform a whitening linear transform that renders these RDM estimation error distributions isotropic. And after doing that, we can trust the Euclidean distance or other simple ways of comparing RDMs, such as the cosine similarity or the Pearson correlation coefficient. So after we do this, we end up correctly choosing model 1 here as the better model for explaining the representation in this data set. And this has been worked out, so we know how to estimate this dissimilarity estimation error covariance by W1D3 and Heiko Schütt, and it's implemented in our open source Python toolbox, so you can check it out there or play around with it in the tutorials. So this gives us whitened RDM comparators, and there's a little more under the hood, so we can use this knowledge about the covariance of the RDM estimates in different ways. We can use it in the context of biased RDMs. For example, we can use biased RDMs and the cosine similarity, and this turns out to be equivalent to the linear-centered kernel alignment, which is a popular way of comparing representations in machine learning. But it's more general than that. So we can also use the Pearson correlation in the context of biased RDMs, or we can use unbiased RDMs, so these unbiased cross-validated distance estimators, and then use the cosine similarity or the Pearson correlation and have whitened RDM comparators for this context. All right, let's talk about step 3. Step 3 is model comparative inference. So now we're talking about statistical inference comparing multiple neural network representations as models of a brain representation. So we want to compare these models. We think maybe some models that are better models of a representation in the brain than others. And we hope through these comparisons to gain insights into the computational mechanisms underlying brain computations. So when we make predictions from neural network models, we often want to fit some parameters. For example, we might not be sure, given a model representation with a set of tuning curves, how prevalent these different tuning curves are in the neural population that we've measured. So some fitting is often desirable, and where there's fitting, there's overfitting. And so in order to prevent the overfitting from biasing our performance estimates, we have to do cross-validation. So we designate a set of fitting stimuli and a set of test stimuli, so two disjoint sets of stimuli, and we also designate two disjoint sets of subjects, the fitting subjects and the test subjects. And then we fit our models using only the fitting stimuli and only the similarities estimated from fitting subjects. Then our models are ready for predictions, and they make predictions for the test subjects RDM for the test stimuli. So the models must generalize simultaneously across subjects and across stimuli, which is a hard generalization challenge. And we can do this in cross-validation to get unbiased estimates of model performance. So it's good to have these unbiased estimates of model performance, but we still need to also estimate our own uncertainty about these estimates. And therefore, we wrap bootstrapping around this cross-validation procedure. And the bootstrapping is also two-factor bootstrapping. So we bootstrap resample the subjects as well as the stimuli in order to simulate how variable our estimates would be if we repeated our experiments for different random samples of subjects and stimuli drawn from the same distributions. So the model comparative inference uses two-factor cross-validation to avoid overfitting to either subjects or stimuli, and it uses the two-factor bootstrap wrapped around the cross-validation to treat both subjects and stimuli as random effects for the purposes of inference. And we think that this way we can get model comparisons where when one model is significantly better than another model, we can actually trust that. We can think that if someone replicated our experiment with a different sample of subjects and a different sample of stimuli, then they might get the same result. So the model evaluation gives us something like this bar graph, which shows the performance of different models as representational models of a reference representation, for example, from a brain. This is simulated data, but this is what it looks like when you compare different predicted RDMs to brain RDMs measured in subjects. And what is plotted here is the RDM prediction accuracy, and here it's the mean across the subjects of the Pearson correlation between the predicted RDMs and the measured RDMs. Importantly, we get these indications here of significance. So at the top here, you see what we call Golan wings, which show the significant pairwise differences. So this is an exhaustive summary, visual summary of all the possible pairwise model comparisons. And the way this works is you look at the dot here, that's the reference model. So this would be FC6 in this case. And then there's a little tick mark for each other model that this reference model significantly dominates. So it significantly dominates conf5 here. It doesn't significantly dominate FC6, but it does significantly dominate FC8 in this case. And then importantly, here's the noise ceiling, which has an upper bound and a lower bound, and it gives us an estimate of the range in which we would expect the true model, if it were one of our models, to fall in terms of performance, given the noise and the intersubject variability in our data. Below the lower bound of the noise ceiling, we have these dewdrops here, which indicate significant tests against the lower bound of the noise ceiling. And you can see that these dewdrops are present for all of the models. So all these models fall short of explaining the data, except conf2 here has evaporated this dewdrop because it comes quite close to the lower bound of the noise ceiling. And we also have these dewdrops on the line at the bottom here indicating significant tests against chance performance, which is a very low bar. And all models here explain significant variance in the data RDM in this case. In tutorial 3, you're going to dig a little deeper on statistical inference on representational geometries and try out our toolbox for this purpose. So a take-home message is that we can compare neural network models in terms of their ability to explain the representational geometry of a brain region. And that's an important tool for computational neuroscientists to learn about computational mechanisms of biological brains. So in conclusion, short overview of the tutorials. So in today's tutorials, you go into a lot of the issues that Heiko and I have briefly introduced you to: generalization and representational geometry, computation as transformation of representational geometry. You'll play around with placing the layers of a neural network in a metric space and looking at the computation as a path through that metric space. You're going to try out statistical inference on representational geometries, and you're going to explore representational geometry and noise and see how the error covariance can be used to normalize the data, and you can measure the distances in a way that reflects the discriminabilities, and you can also remove the bias of distance estimates. So have fun over the day. I hope the tutorials will be inspiring and will somehow contribute to your own research work. Thank you very much.